Today we're going to talk about muscle tissue. We're going to go over the three different types of muscle tissue, which include skeletal muscle, cardiac, and smooth. We'll discuss the properties of muscle tissue and the functions of muscle tissue. So the properties of muscle tissue include electrical excitability. That means that it has the ability to respond if stimulated. Contractility means it has the ability to shorten once it's stimulated by an action potential. Extensibility means that it has the ability to stretch without tearing or damage during contractility and when it's stimulated. Elasticity means that it has the ability to stretch out and then recoil back to its resting length. And autorhythmicity means that the stimulus arises out of the muscle itself. We see these in cardiac muscle as the pacemaker and in smooth muscle. So let's look at the three types of muscle that exist. So the first one is called skeletal muscle. So skeletal muscle has long cylindrical fibers. Right here is one muscle fiber. And do you see these little lines? We call those striations. Skeletal muscle histologically also is multinucleated. So the function of skeletal muscle is that it connects to the skeletal system, and when contracts, it'll move the body. Also has the ability to create heat, and it's controlled by the nervous system. So if we look at these striations, the striations are made up of proteins that are called actin and myosin, and that's why they appear striated in this image. Cardiac muscle is only located in the heart. Each muscle cell is called a cardiocyte. So histologically, their cells are smaller than skeletal muscle cells. The nuclei are centrally located. The unique thing about cardiac muscle cells is in between each cell, they have intercalated discs. And what they are regions of gap junctions where the electrical impulse can easily pass from one cell to the next. Cardiac muscle does not divide and repair. And the fact it also has autorhythmicity. So what autorhythmicity means that it can stimulate within itself, and in cardiac muscle, they're called pacemaker cells. The nervous system can alter the speed, but the initiation is within the heart itself via the pacemaker cells. The third type of muscle is called smooth muscle. And smooth muscle is located in a lot of our vessels and tubes. So we see this inside the eye, the respiratory, digestive, urinary, and reproductive tubes. Also the erector pili muscle. Remember, those are the muscles that when contract enhance and cause the sebaceous oil glands to secrete more sebum around the hair follicles. Smooth muscles are small and unbranched. They can divide and repaired, and they are unstriated and also involuntary. So if we look at the summary of what we just talked about, the skeletal muscles are long cylindrical fibers, and they are appearing striation. They primarily attached via tendons to bones. They do not have autorhythmicity, and they're completely voluntary, so we can consciously control this, and they have a limited capacity for regeneration when injured. Cardiac muscle cells are branched. They're smaller. They're only located in the heart. They are striated, just like skeletal muscle, but they're only one nuclei per cell. So the cardiac muscle does have autorhythmicity. It's totally involuntary. They're controlled by the autonomic nervous system and limited to none ability for regeneration. The smooth muscles taper off at the ends. They have a centrally located nucleus. And this one are completely involuntary. And the interesting fact about these is that they do have the ability to repair if needed. So make sure you understand histologically what each of these three would look like. So skeletal muscle have these long cylindrical tubes that are horizontal to each other. They have striations, and they also are multinucleated. Skeletal muscle is under voluntary control, and they're responsible for voluntary movement. Cardiac muscle are branched. They're smaller. There's only one nuclei per cell. They're also striated, as we can see in here. They're found only in the heart, and they're involuntary and have autorhythmicity. Smooth muscles, the cells are tapered, kind of connected to each other in this manner. Totally involuntary. It's found in the walls a lot of our internal organs, such as intestines, tubes, and such as blood vessels. They're totally under involuntary control, and they do have autorhythmicity. So let's look at the structure of skeletal muscle. We'll focus in on this type of muscle in this section. So when we look at a skeletal muscle, we notice that it has a tendon, and a tendon connects to the periosteum of bone. So it's a very elastic connection. So therefore you could pull on it, and the ability for it to tear is minimal. So it has elasticity and extensibility. Then we have the muscle belly. Okay, so the way the muscle connects to the bone is via the tendons. If we look around the whole outside, there's a fascia, which is a dense, irregular connective tissue that kind of separates each muscle bag, kind of in a bag-like separation. When you see the term aponeurosis, this means a large tendon that it could either connect to another muscle, a bone, or the skin. So let's look at the details of muscle anatomy. So here is our muscle. It's connecting to the periosteum via the tendon. So we'll learn that skeletal muscles structurally is divided by connective tissue. So here's the entire muscle tendon, and the connective tissue around that is around the whole outside here is called the epimysium. We'll learn that the epimysium surrounds these bundles of muscle cells, which are called fascicles. So you see each of these circles here? That is one of these. And the fascicles contains tubes of muscle fiber. When you see fiber, muscle fiber, this is a muscle cell. So then if we look inside this fascicle in greater detail, then we'll look inside. The connective tissue around the fascicle is called the perimysium. And then within each of these muscle cells, there's another group of individual little tube called the muscle fiber. The connective tissue around the muscle cell is called an endomysium. So let's look at this in greater detail. So the organization of skeletal muscle cell is in 3 layers. The interesting fact about all of these connective tissues eventually all unite at the tendon, and therefore this tendon is very, very strong with elastic and its elasticity and extensibility. It's very difficult to tear, not that it cannot, but it's difficult to tear a tendon away from its bone. So again, the epimysium surrounds the entire skeletal muscle belly. It's made up of, it also surrounds these little almost bags of muscle cells, and that connective tissue around each of these fascicles is called the perimysium. So up until this level, you could see skeletal muscle with the unaided eye. If you've ever eaten steak or meat, the little fibers you see when you're, cutting that is up until this layer. Once we get to this layer, this is microscopic anatomy. So each muscle cell here has its connective tissue around that, and that's called the endomysium. So let's look at this in greater detail. So around the whole outside of the cell is the epimysium. It's made up of dense, irregular connective tissue, and it binds all the fascicles together. These are fascicles or bags of muscle cell or muscle fibers. So again, all of these connective tissue, that includes the epimysium around the entire muscle itself, the perimysium, which is the connective tissue around the fascicle, which is made up of muscle cells, little tubes of muscle cells. Then the connective tissue around one of these tubes is called the endomysium. So if we look at the entire muscle cell, that's the epimysium. Each of these little bags here are called fascicles. The connective tissue around the fascicle is called the perimysium. So within the perimysium, it contains collagen and elastic fibers and blood vessels and nerves that bring all of this to each fascicle. And within the fascicle, it's made up of little tubes of muscle fibers or muscle cells. And the endomysium is the connective tissue around one of these individual cells. So here we are, the fascicle. We've got the endomysium around one of these muscle cell or muscle fibers. So the connective tissue of the endomysium surrounds each muscle cell. And when you see muscle fiber, it's the same thing as the muscle cell. So these are loosely interconnection of fibers. They contain capillaries and satellite cells, which aid in limited repair if damaged, and also contain nerves. So it's important to understand the organization of skeletal muscle, going from the largest, which is the epimysium around the entire cell, perimysium around each fascicle, and then the endomysium around each muscle cell fiber. So this is from your textbook. Make sure you understand the organization of skeletal muscles. So we start with the skeletal muscle itself, and it's the connective tissue around that would be the epimysium. And then this is the epimysium surrounds these bags called fascicles, which contain bags of muscle cells. The connective tissue around the fascicle is called the perimysium. So then if we look at one of these muscle cells, this is where we get into microscopic anatomy. Everything from here up we could see with the unaided eye, but everything from here down we need a microscope to see this. So this is a muscle cell, which we'll talk about in much more detail. The muscle cell is made up of little tubes called myofibrils. Myofibrils are made up of filaments, myofilaments, and these filaments are either thick or thin filaments, and we'll get into this in a little while. So let's review everything we've covered up to date. So we've discussed in the past that the outer layer is called the epidermis. The middle layer is the dermis of the integument system, and then the subcutaneous layer is part of the integument system, but not part of the skin. If we pull that off, fascia is a layer of dense irregular connective tissue that covers the muscles. It kind of separates each muscle. If we take that off, the muscles off, this is a tendon here, then we get to the periosteum of bone. The periosteum is that outer layer of dense irregular connective tissue. This is the tibia, and then within the tibia there's a medullary cavity. We talked about how between the tibia and the fibula there's the interosseous membrane, a form of connective tissue. Fibula is the bone. Tendon is the way a muscle connects to the periosteum. The retinaculum it's a band that wraps around tendons and holds them in place. So let's continue with microscopic anatomy. So this is a muscle cell, correct? And the connective tissue around this entire muscle cell would be the endomysium. So the muscle cell is made up of myofibrils. So the muscle cell is made up of little tubes called myofibrils. And then within the myofibrils, there's even smaller little tubes called myofilaments. So we'll get into all of this in greater detail. But let's kind of look at what we're seeing here. So the myofibril is actually the contractile element of skeletal muscles, and we'll look into how this occurs in a minute. Some other things I want you to note around the muscle cell is these purple little bands around the myofibril. They're called sarcoplasmic reticulum, or SR. They're fluid-filled membranous sacs, and they're similar to our original cell, which was the smooth endoplasmic reticulum. But in our skeletal muscles, the sarcoplasmic reticulum store calcium. And we'll learn that calcium is needed to initiate muscle contraction. So calcium is stored in the sarcoplasmic reticulum until stimulated to trigger this release out of the terminal ends of the sarcoplasmic reticulum, which are right here, and they're called terminal cisterns, okay? You also see these blue tubes, and they're called transverse tubes. So if we kind of step back a little bit, and we notice on this model, the sarcolemma is the plasma membrane of our muscle cell. The sarcoplasm is this material in between the myofibrils, and this contains myoglobin, which is an important O2 binding protein. You see in the cell here, there's mitochondria. The mitochondria in our skeletal muscles have the ability to reproduce themselves, especially if we have a greater need for more oxygen production or energy production. So the other thing I want you to note here are the transverse tubules. So the transverse tubules are actual extensions of the sarcolemma. And then note what a triad means. So a triad is this region here, which includes the two terminal cisternae and the transverse tubule. We call that little system the triad. So let's look at our muscle fibers or muscle cells. So the muscle fibers in general, we're born with all the muscle cells we'll ever have. So most growth in these cells after birth occur due to hypertrophy, meaning the cell itself grows, versus hyperplasia, which occurs in utero, where we're increasing the number of fibers. So if we look in the muscle cell itself, it's made up of these myofibrils, which are made up of myofilaments. And those myofilaments, there's a thick filament and a thin filament, which we'll discuss these proteins shortly. So these filaments are arranged in these compartments called sarcomeres, and the sarcomeres are considered the smallest contractile unit of skeletal muscles. And these patterns of thick and thin filaments is what makes the pattern of striations when we look at skeletal muscle under a microscope. So we talked about the sarcoplasmic reticulum. So you can see in blue here, this is the sarcoplasmic reticulum. It's similar to the smooth endoplasmic reticulum in our cell, and they store calcium in the sarcoplasmic reticulum. Calcium is necessary in order to initiate muscle contraction. So they're stored in the sarcoplasmic reticulum, and they're released at the region of the terminal cisternae when stimulated. The sarcoplasm is the cytoplasm of the muscle cell, and within this we have glycogen that's stored in here that's used for rapid energy source, and myoglobin is located in the sarcoplasm, which is a protein only found in muscles that binds to oxygen. So also we'll move on to look at the sarcolemma. So we talked about how the sarcolemma is the plasma membrane of our skeletal muscle cells. And what the transverse tubules are, which we've talked about in the past, these little tubes, they're invaginations or infoldings of the sarcolemma. So along the sarcolemma, the action potential stimulates the sarcolemma, and this impulse will go immediately throughout the myofibril and deep in the muscle cell via these transverse tubules. So they're filled with interstitial fluid, and this helps propagate the action potential throughout the entire muscle cell quickly. So this stimulation or the action potential that's going through the transverse tubule will stimulate the sarcoplasmic reticulum to release calcium around the myofibril through the terminal cisternae, which is part of the sarcoplasmic reticulum. So again, make sure you note the triad includes the terminal cisternae plus the transverse tubule. So it's one transverse tubule, two terminal cisternae equals the triad. So let's look at this myofibril in greater detail. So first of all, they run longitudinally and through the entire muscles and they contain sarcomeres. Do you see this is a sarcomere? So we'll talk about, in general, the myofibril is a contractile element, and the sarcomere is the smallest contractile element of skeletal muscles. So the myofibrils make up most of the muscle volume. They're arranged in little filaments. See these little circles that are either thick or thin proteins, and we call them thick or thin filaments. The sarcomere, the ends of the sarcomere are divided by these little lines called Z-discs. So from here, Z-disc to Z-disc, that's considered one sarcomere, and then the next sarcomere would be from this Z-disc to the one out here. So the myofibrils, they have this pattern of overlap between thick and thick filaments, and that's what gives the skeletal muscle this striated pattern that we see, the thin filaments here and the thick filaments here. So let's look at this sarcomere. This is considered the contractile unit in striated striated muscle cells and extends from one Z-disc to the next. So let's look in detail what all of this means. So this is the sarcomere. We know it's the sarcomere because it goes from Z-disc to Z-disc, okay? So if we look in this image here, we could see the filaments. So one of these little circles here is one of these filaments. And you could see the myofibril, or specifically this little section called the sarcomere, is made up of thick filaments, which is the orange, which connect to the center line called the M line, and thin filaments, which you can see here in yellow, that connect to the Z-discs on each side, okay? So these are considered the thick filaments in orange, and the yellow is considered the thin filaments. So other lines we have to know if we're looking at this is, first of all, Z-disc to Z-disc contains the sarcomere. This lighter pink area is the area where we only have thin filaments. This region in here is a region of overlap, and the little section in the middle, or the H zone, only contains the thick filaments. So let's look at that again. So this H zone, which is this center zone here, which would be right in here, okay? That only has thick filaments. Do you see? You only see the orange. The A band, which would be this region here, which you could see right here, only is the region of overlap, the overlap of thick and thin filaments. The I band, which is this zone here, so it would be part of the next sarcomere that extends this way, which you could see right here. So the I band, as you could see, only has thin filaments. So let's review this. The Z-discs separates one sarcomere from the other, and it's this repetitive units of sarcomeres that run along the whole longitudinal tube of the myofibril. The A band, which you could see right here, the A band is the darker middle part of the sarcomere, and it's the area of overlap between thick and thin filaments. The I band, and the way we're going to remember that, I is thin as opposed to H is thick. So I band only has thin filaments, where the H zone is in the middle here, only has thick filaments, okay? And the M line in the center connects to the thick filaments, and the Z discs connect to the thin filaments, plus they separate one sarcomere from the other. So if we look at this on an advanced microscope, we could see this pattern where here is the Z discs, right? Z disc to Z disc. So this region, this whole region, is that sarcomere. So the region of overlap, which is right here, it's a darker area in the center. This is the A band, or the region of overlap. This section in here, which is that middle section, is the H zone. And what did we learn about H? It's thicker than I, so it's only thick filaments. And then here is another sarcomere, and this is this sarcomere. That is considered the I band. And remember, the I is thin, only has thin filaments. Okay? So let's review it again. We could see the sarcomere is right here, right? Which is the functional contractile unit of the muscle. The M line is the center, and the M line connects to these thick filaments. The A band, which is this region, is the region of overlap. The I band here has the green fibers in it, only has the thin filaments. And we'll talk about different proteins that stabilize these filaments in place. So proteins connect to the A bands and the I bands, the dark bands and the light bands, and these proteins, we'll discuss their details in a minute. So the M line is that center line that connects to the thick filaments. The H zone is this lighter region in the middle, but this only has the thick filaments, and the A band is the region of overlap of thick and thin filaments. So we know the sarcomere extends from Z line to Z line, and we'll discuss the types of proteins that exist in all of these thick filaments. And titin is a specific protein that connects the thin filament to the Z line and connects the thick filament to the H line. So here's a review of what we talked about. Make sure you know all of these names and what they do. So again, the Z discs separate one sarcomere from the other. The A band is the region of overlap. The I band only contains thin filaments. The H zone only contains thick filaments. The M line is the center that connects to the thick filaments. So let's talk about these muscle proteins. So the myofibrils are built from three types of proteins. One's a contractile protein, and their myosin is one of the major thick filaments, and actin is one of the major thin filaments. These are contractile proteins. Then we've got regulatory proteins, and they're called tropomyosin and troponin, are part of the thin filaments. We'll look at images of this. So they're called regulatory proteins. And then the third type are structural proteins. The main one is titin. This anchors both the thick filament to the Z discs and the thick filament to the M line and the thin filament to the Z disc. They assist in elasticity and extensibility. There's other proteins that exist throughout, but the main one, main structural one that we see a lot is the titin protein. So let's talk about the thick filament. The thick filament is made up of the protein myosin. So the myosin thick filament kind of looks like a golf club wrapped around itself. So when we look at the thick filament is made up of a head and a tail, and they're kind of twisted around each other, and this is what they look like histologically. So the head and the tail with the thick filament. The thin filament is made up of actin as a contractile protein, which is the blue. Then we've got two regulatory proteins. One is troponin and one is tropomyosin. So they're considered regulatory proteins. So in this position, they block the myosin head from connecting to the actin head. So that's why we call them regulatory protein. So here's the thick filament with the myosin heads. So in order for contraction to occur, it's almost like the thick and thin filaments walk over each other as contraction occurs. So here's the thin filaments with the actin. We've got the troponin and the little bands of tropomyosin. So it's in a relaxed state, the tropomyosin prevents the connection of the myosin head with the actin. So as we talked about, when contraction occurs, it occurs along the sarcomere, and it's not like the sarcomere itself gets shorter. It's the thick and thin filaments walk over each other. Here's the Z lines, okay? So the Z lines get closer to each other as the actin and myosin head walk over each other. And that's the basis of the sliding filament theory. So when this occurs, several things can be seen. First of all, the I bands are going to move over the A bands, and the length of the thick and thin filaments is going to remain unchanged. So let's look at this. So each one of these here is the sarcomere. Okay, so here's Z discs to Z disc here. So this is in a relaxed state. You could see the Z discs. We could see in this case the A band, which is the region of overlap. We could see the I band, which only has the thin filaments. And this is the H zone in the next sarcomere, and this is the region where it only has thick filaments. So as contraction occurs, the Z discs are going to get closer to each other. So what things remain the same and what things are going to disappear? So the H zone here, because these walk over each other, this is going to disappear. So the H zone is going to disappear, right? And what was in the H zone? What was located in this H zone is the thick filaments. The I bands also get smaller and it disappears. So the I bands are going to disappear, right? So the I band, which has what? Only thin filaments. How about the A band? What was the A band, the region of overlap? That's going to remain the same because they are overlapping each other. What about the length of the thin and thick filament? That's also going to remain the same because contraction occurs with what we call the sliding effect of the thin over the thick. So what happens is the myosin head connects to the actin protein, and they walk over each other as contraction occurs. So therefore, the A bands get smaller, the zones of overlap become greater, and the Z lines move closer together, or the length of the sarcomere. The width of the A band is going to remain the same, and this sliding filament theory explains how the myosin head walks over the actin as muscle contraction occurs. So the I bands disappear, the H bands disappear, the A band remains the same, and the length of the thick and thin filaments remains the same. So when we talk about the entry of nerve and blood into our muscles, they usually enter the muscle at the tendon origin. We'll learn that there's an origin and an insertion in the muscles, and the entry of this group is called a neurovascular bundle, which allows the nerves and the vessels to enter the muscle tissue. We've talked about before how nerves could either be motor, efferent, or sensory nerves. And they enter together called the neurovascular bundle. You can see this grouping here is called the femoral artery, nerve, and vein. When we reach this area, this mnemonic is called VAN. So we discussed motor neurons in the past. So remember, the motor neurons are the reaction. So once the central nervous system is telling our skeletal muscle to contract, this information is carried via a motor neuron. So where a motor neuron connects to a muscle cell, this connection is called the neuromuscular junction. So let's talk what happens at the neuromuscular junction. Remember from the past, we talked about a neuron and the axon terminal, and the synapse is where the communication occurs, where a motor neuron is communicating, in this case, with a muscle cell, and that's called a neuromuscular junction. So the action potential is reaching the axon terminal, correct? It's going to stimulate the synaptic vesicles to release via exocytosis their neurotransmitter called acetylcholine. So the neurotransmitter is a chemical messenger, and this messenger goes into the synaptic cleft, and then within the synaptic cleft, it binds to the neurotransmitters, which then open up the ion channels, allowing that electrical impulse to get into the skeletal muscle cell. And this is located along the sarcolemma. The part of the skeletal muscle cell, which is the receiving end of this, is called the motor end plate. So let's continue to see what happens. So again, the action potential reaches the synaptic end bulb. This stimulates the vesicles to release acetylcholine from the axon motor neuron, crosses the synaptic cleft, and then binds to the acetylcholine receptors on this receiving portion of the skeletal muscle cell is called a motor end plate. This in turn stimulates the action potential along the sarcolemma, okay? And we already talked about how the sarcolemma is connected directly to these transverse tubules, okay? These are the transverse tubules. And it's an interesting occurrence. So this is the action potential immediately going through the deepest layers of the myofibril. Here's our sarcoplasmic reticulum here, okay? And what's stored in there? It's calcium. And see these ends here are the terminal cisternae, which are part of the sarcoplasmic reticulum. So let's backtrack here. At that neuromuscular junction is the connection between the motor neuron coming from the central nervous system, that impulse where it connects to a muscle. So at this junction, acetylcholine, which is the neurotransmitter, crosses into the motor end plate of the muscle cell. And what happens here is this produces an action potential in the receiving myofibril of the muscle. So this action potential triggers an electrical impulse which runs along the sarcolemma. And the sarcolemma is connected directly to the transverse tubules, which stimulates the sarcoplasmic reticulum to release calcium into the sarcoplasm. And this calcium is released through the transverse tubules. So this process is broken down when we release an enzyme to stop this process from occurring, and will allow the muscles to relax. So when we talk about muscle contraction, we talk about this sliding filament theory. So because calcium is present and ATP is present around the skeletal muscles, it allows this calcium that is released from the terminal cisternae into the sarcoplasm of the myofibril. So then we have the calcium present within the areas of the thick and thin filaments. So what happens at this point, we need energy, ATP, and we need calcium present. So what happens is that the calcium binds to the troponin, which pulls the tropomyosin out of the way, allowing the myosin head to connect to the actin head. So once this connects, it begins to pull towards each other. They slide over each other, and therefore the Z discs come closer to each other. The hydrolysis of ATP to ADP and phosphate also powers this cyclical process. So calcium, when calcium is removed back into the sarcoplasmic reticulum, the tropomyosin blockage is restored and the muscle can relax. So again, here's our thick filament, which is the myosin, and here's the myosin head. So when calcium is present, first of all, we need energy. ATP is broken down to ADP and phosphate, and this activates the myosin head to connect to the actin. Also, the calcium that is released into the sarcoplasm from the terminal cisternae, which is part of the sarcoplasmic reticulum, that binds to troponin, which pulls the tropomyosin out of the way and also permits this myosin head to connect to the actin. And also we need the release of ATP. So once they connect. What occurs is a power stroke where it kind of walks over each other, and the myosin head binds to ATP again and it becomes inactive. So it continues this process of contraction until calcium is no longer present and it will relax. So again, let's look at this. First of all, the myosin head hydrolyzes ATP and it gives it the power to attempt to connect to actin. So it does connect to actin as long as calcium is present, and that binds to troponin, pulling the tropomyosin out of the way. So the myosin is going to cause a power stroke or a walking over that area. And then once calcium is not present anymore, then this separates from the cross bridge, and then the detachment of myosin from the actin therefore allows the muscle to relax. Another theory is called the excitation-contraction coupling, which we talked about the role of calcium in the regulation of contraction. So again, the action potential is going to reach that synaptic end bulb of a motor neuron. So this is going to trigger excitosis along the synaptic cleft, and the ACT will bind to the ACT receptors. So this is allowing the inflow of sodium, which will initiate the muscle action potential along the sarcolemma of our muscle cells. And what this does, it's going to immediately go through the transverse tubules, which will cause the release of calcium from the terminal cisternae. And then as we talked about the presence of calcium within the sarcoplasm will connect to the troponin, which pulls the tropomyosin out of the way, and then it permits the actin and myosin heads to connect, which they slide over each other until calcium is no longer present and muscle relaxation occurs. So let's move on to the types of skeletal muscle fibers. There's three types of skeletal muscle fibers. There's slow oxidative, fast oxidative, and fast glycolytic fibers. So most skeletal muscles vary in how much of these fibers exists. So red muscle fibers have a high content of myoglobin versus white muscle fibers have less myoglobin in it. So often the ratio of all of these sometimes can be affected genetically, sometimes can be influenced by a person's physical performance. So again, slow oxidative fibers, which are type 1 fibers, they're the smallest. We've got the most of these. So they have high capacity for aerobic cellular respiration. So they have a high fatigue resistance, and they're considered our postural muscles. They're muscles that are, like, for example, for me to be standing in front of the room lecturing for an hour or two, these are the postural muscles holding my neck in place. So they're maintaining posture, and they have also aerobic endurance activities. Fast oxidative fibers are intermediate. They're kind of a combination of red and pink color. So these muscle fibers have an intermediate capacity, having both aerobic cellular respiration and anaerobic cellular respiration or glycolysis. So they're considered muscles, for example, that allow us to walk or sprint. The fast glycolytic fibers, they look white if we look at them. They're muscles that have low capacity of anaerobic cellular respiration. And they're more rapid, intense movements of short duration. For example, like weightlifting. So let's look at other terms. As we start memorizing the names of muscles, we have to know certain terms. An important one is origin and insertion. And we also have to know the actions of all our muscles. So we know that skeletal muscles cause movements by connecting to the bones, the skeletal system, via tendons. So the way they connect to the bones is the way we describe them. So when we see origin, this is the attachment of the muscle via the tendon to the stationary proximal part of it. So the origin does not move. The insertion is the attachment of the muscle via the tendon to the part that moves, the mobile part. And the action means what movement occurs when the muscle contracts. So again, here's our biceps brachii. The origin, actually it's biceps, it has two origins, is first of all, the coronoid process of the scapula is one origin, and also the other origin comes from the glenoid fossa, the supraglenoid tubercle. So these are the origins. They do not move. They insert into the radius, and this is when this muscle contracts, what it's going to do is going to produce elbow flexion. So the origin are the origins of the biceps brachii at the region of the coronoid process, and also the supraglenoid cavity of the glenoid fossa or glenoid tubercle. And the movable end is along the radius. And the action is elbow flexion. So origin is the immovable part of the muscle attachment. The insertion, attachment of the movable part. So make sure we understand the difference. The origin does not move, the insertion moves. So more complex movements that we have to understand are agonist, antagonist, and synergist. So the agonist is considered the prime mover. So if we go back to this, biceps brachii, the prime mover of elbow flexion would be the biceps brachii muscle. So it's the muscle whose contraction is primarily responsible for producing a movement. So the agonist would be the biceps brachii when we're talking about elbow flexion. So in order for the agonist to function, often there's an antagonist. So this muscle, which is the triceps, which is located in the same region of the biceps brachii, but on the posterior region of our upper arm, and that's called the triceps. So in order for elbow flexion to occur and the biceps brachii to contract, the triceps has to relax in order for this to occur, and this relationship is called agonist and antagonist. A synergist muscle is a muscle that aids the agonist. For example, in this situation, the biceps brachii, there's a muscle called the brachioradialis, which acts like a helper to the agonist. So that would be called the synergist. So again, let's look at this image. The prime mover in this situation would be the biceps brachii when we're talking about elbow flexion. There's a muscle in this region that connects the humerus to the radius. It's called the brachioradialis. This is the helper muscle. It reinforces the same movement, also stabilizes the movement, called a fixer. So in order for this to occur effectively, we need an antagonist, and that would be called the triceps. So in order for this movement to occur effectively, elbow flexion, the triceps muscle has to relax, and that's considered the antagonist. So we learned that kinesiology is the study of motion. And let's talk about a lever system that exists with the body. So let's talk about some definitions. The fulcrum of a joint is a fixed point. We talk about the effort. It's the cause of the movement. So the force caused by a muscle contraction, when the muscles contract, cause a movement at the insertion. The load will be the effort that we put on the movement. The resistance opposes the movement. There's a difference between mechanical advantage, where when the load is closer to the fulcrum, there's less effort needed to move. And there's a mechanical disadvantage when the load is further away from the fulcrum, a larger amount of effort is needed. So let's look at the three types of lever system that exists. So there's type 1, first class, second class, and third class. So first class would be like a scissor, and this is an example of, for example, our cervical spine. So first of all, the fulcrum is along the spine itself. The effort is the cervical muscles, and the load would be the weight of our head. And that's called the first class lever. The second class would be like a wheelbarrow. So in this case, the load is going to be the weight. The effort would be the muscles that have to contract. And the fulcrum, for example, would be like standing on our toe, or the front part of your foot. The third class is the most common one. Okay, so the third is the most common type of lever system, and this is more where the effort, which in this case would be the muscle, the effort is applied between the fulcrum and the load. An example would be forceps. So the fulcrum would be the elbow joint, the load is how much we're lifting, and the contraction of the muscle would be the effort. So the third class is the most common lever system in the body. Other terms when we're talking about muscles includes the difference, knowing the difference between isotonic and isometric contraction. So when we think of isotonic, this means that the tension within the muscle remains almost the same while the muscle changes in length. And there's two types of isotonic, meaning the tone remains the same. There's concentric isotonic versus eccentric. So concentric, for example, in elbow flexion, we're lifting a weight, is the movement of flexion, and the eccentric is the lowering of the weight and muscle lengthening. Isometric contraction means that the tension is generated is not enough to move the muscle. And let's give you an example of this. So an isotonic contraction, as we contract, like for example, as we contract our biceps brachii to lift the ball, versus isometric, if we're just lifting a weight out in space, our muscles still have to contract and relax in order to keep it. There's no movement in the muscle. So isotonic, concentric, for example, weightlifting, we're moving it closer. Eccentric is elongating of the muscle, but controlling it as we as we release the weight. Isometric contraction, it's, for example, holding this weight in the air. If you ever tried something like that, you would realize how much strength you have to have this. That is considered isometric contractions. So that is the end for today.